Basically, we're talking about an extensive system that consists mainly of the epithelium and underlying structures of the digestive tract, as well as its associated glands. The gland we'll concentrate the most on, obviously, is going to be the liver. First off, let's illustrate in 2 slides, one that's in the notes, one that we incorporated into this video, that illustrates the fundamental histologic similarities in terms of layers of the digestive tract. Innermost and closely associated with the lumen of different components of the digestive tract is the so-called mucosa, and the mucosa of the GI tract consists of a lining of epithelium, which we'll see is mostly going to be a simple columnar epithelium, an underlying vascular and connective tissue, lamina propria, and immediately deep to that will be a layer of muscle known as the muscularis mucosa. It is in the area of the mucosa where we see the most distinct differences in various components of the GI tract. Underlying the mucosa will be an area of submucosa, then in one region of the GI tract, the duodenum, which will contain some prominent glands. Underneath that are circular and longitudinal layers of the muscularis externa, basically consisting of smooth muscle. And in various parts of the GI tract, there may be a serosa or peritoneal covering, either completely or virtually completely covering a layer or a portion of the GI tract that's peritoneal, or only partially covering a portion of the GI tract if it is secondarily retroperitoneal. So now a better picture that we borrowed from Kierszenbaum's Histology and Cell Biology text basically serves to illustrate all of the different structural features in one picture of the regions of the GI tract, beginning with the esophagus and going down to the area of the large bowel of the colon. And again, you can see, as illustrated here, and then we'll underscore this in chart form, the major histologic differences in regions of the GI tract come in different parts of the mucosa. And again, you can see fundamentally, as we illustrated previously, the structural layers of the GI tract begin with a mucosa, which is epithelium plus underlying lamina propria, plus a thin layer of the muscularis mucosae. Underneath that, a submucosal layer that may or may not have some glands in it. And then the muscularis layer consisting of longitudinal and circular layers of smooth muscle. And then finally, an outer layer of adventitia, again connective tissue, that may or may not be in close contact with an overlying layer of serosa, which is the peritoneum. So let's go through these histologic layers and differences and then underscore them with the specific cell type features that are in the charts on the subsequent couple of pages. First off, note that the esophagus is distinct in terms of its epithelium because it's the only part of the endoderm lining of the gut tube that's characterized by a stratified squamous, non-keratinizing epithelium. Basically, other than that, the histology of the esophagus is unremarkable except for the presence of some underlying submucosal glands that open periodically into the lumen of the esophagus. Because in a way, certainly the esophagus is merely a conveyance tube to get the bolus of food from the pharynx down to the stomach. The stomach epithelium begins our tour of epithelium that's basically thrown into pits and deep and shallow gastric glands, all lined by different functional elements of a simple columnar epithelium. Other than that, the underlying lamina propria and muscularis mucosa layer are unremarkable. Once we get down to the duodenum, the simple columnar epithelium is largely absorptive, highly microvilliated, and throughout the length of the small intestine, duodenum, jejunum, and ileum, the lamina propria and the overlying epithelium are thrown into distinct finger-like projections known as villi, which certainly serve to increase the surface area available for absorption of these 3 regions of the small intestine. In the duodenum, we'll be able to distinguish a duodenum section not by its epithelial and underlying lamina propria characteristics, because all parts of the large and small intestine not only have a simple columnar epithelium, but as you can see, all parts of the small and large intestine are characterized by the pronounced invaginations known as crypts of Lieberkühn. They're not actually labeled on this, oh yeah, they are labeled on this particular diagram, as illustrated by those white lines projecting down deeply into the lamina propria. The duodenum is distinct from jejunum and ileum by the presence of the submucosal Brunner's glands, which are producing an alkaline secretory product to counteract the acidic medium that's being presented to the duodenum from the lumen of the stomach. The jejunum and ileum certainly are going to lack the submucosal Brunner's glands, but the way you can distinguish histologically the jejunum from the ileum is by the presence of prominent patches of B lymphocytes in a Peyer's patch situated immediately adjacent to the luminal surface in the underlying lamina propria. So the fundamental distinctions between areas of the small intestine are this. The epithelium is simple columnar absorptive thrown into folds or villi, and basically crypts of Lieberkühn characterize all parts of the small intestine. But in the duodenum, the submucosal area will have Brunner's glands. In the ileum, the underlying lamina propria will be characterized by Peyer's patches. In the absence of both, obviously the indications are that you are in a section through the jejunum. The colon is still going to have the simple columnar epithelium, but it's only going to have the crypts of Lieberkühn because the villi will now be basically absent. So now in chart form on the next page, we can summarize these differences with a kind of a look at some of the specialized cell types that are found in various areas of these regions of the gut tube. Certainly, as we've indicated, the esophagus is the only part of the endoderm lining of the gut tube that has a non-keratinizing stratified squamous epithelium, which we've underlined as indicated here. Differences in the muscularis portion of the esophagus is the transition that takes place from skeletal muscle in the upper third to completely consisting of smooth muscle in the lower third. Basically, the middle third of the esophagus probably has a mix of both skeletal and smooth muscle. The skeletal muscle in the upper portion of the esophagus is being innervated by basically skeletal motor neurons in the vagus nerve, whereas the skeletal muscle in the lower portion of the esophagus is innervated by autonomic nerve fibers, also carried by the vagus nerve, which obviously answers the innervation question that's raised here at the top of this chart. By the time we get to the stomach, basically now the major characteristics of the epithelium are simple columnar. The body and the fundus of the stomach are thrown into shallow pits and deep gastric glands. The characteristic cell types are numerous in the body and the fundus of the stomach, consisting of a variety of mucus cells that serve to protect the corresponding epithelium from the acidic medium that's in the lumen of the stomach. Also found at various points down at these in these deep gastric glands are chief cells that produce pepsinogen and lipase precursor. They tend to be found at the bottom of these glands. Parietal cells, a variety of locations, both near the luminal surface and more deeply placed. Parietal cells, their major job certainly is to produce hydrochloric acid, but their other major secretory product, which is essential for life, which enhances, obviously, or enables the absorption of vitamin B12 from the terminal part of the ileum, is intrinsic factor. And then certainly there are going to be a variety of enteroendocrine cells that are populating these gastric pits. As we move down to the pyloric portion, we basically have the same array of cell types, but most importantly, this is where we find a concentration of enteroendocrine cells that are producing gastrin. Gastrin's major role is to induce parietal cells to increase their secretory output of hydrochloric acid. As we move now down to the portions of the small and large intestine, as we alluded to earlier, we certainly can say that a common feature of all parts of the small intestine, duodenum, jejunum, and ileum, is the presence of the absorptive simple columnar epithelium, but basically the characteristic features are that all parts of the small intestine are thrown into prominent villi, folds, and interdigitated with crypts of Lieberkühn. So we have to use other criteria histologically to distinguish a section of duodenum from jejunum and ileum. And as we allude to here, the duodenum basically can be distinguished by the presence of the submucosal Brunner's glands, which, as we alluded to, are producing an alkaline secretion to counteract the acidic medium presented to it from the stomach. And The jejunum, basically also characterized by the prominent villi, crypts of Lieberkühn, and so on and so forth. But the ileum can be distinguished from the jejunum by the presence of the patches of lymphatic tissue in the form of Peyer's patch nodules immediately adjacent to the luminal surface epithelium. Down in the crypts of Lieberkühn, in all parts of the small and large intestine are the Paneth cells. Their job is to produce lysozyme, which has a bacterial regulating role. Goblet cells are intermingled among the columnar absorptive cells throughout the length. As we saw in the stomach, the functional significance of the enteroendocrine cells changes, whereby in the duodenum, the EE cells are producing mainly cholecystokinin to basically stimulate the release of bile from the bile duct and the gallbladder, and to basically stimulate the release of secretin as well that increases the output of bicarbonates from the exocrine pancreas. The jejunum, again, same cell types except for differences in the EE cells in the overlying epithelium. The ileum, as we said, has the Peyer's patches, and basically overlying the Peyer's patches are squamate or slightly dome-shaped M cells or microfold cells. These are antigen-presenting cells that basically take up and transport antigen from the lumen of the ileum and present it to the underlying B cells that will generate germinal centers in the underlying Peyer's patches. Large intestine, same basically histological features in terms of its epithelium, but now we lack the villi that were prominent features of all regions of the small intestine. The large intestine still has the crypts of Lieberkühn, and obviously the simple columnar epithelial cells are still mucosecreting and absorptive, but now the absorption basically is involving the active transport of sodium out of the lumen of the large intestine, as well as the passive movement of water down its concentration gradient. Histologic features, now we're looking at a diagrammatic feature of a villus, and you can see its overall lining of enterocytes, the columnar absorptive epithelial cells intermingled with goblet cells. Down at the base of a crypt of Lieberkühn is basically where we find the stem cells that are involved in the regeneration of enterocytes throughout the length of the villus. And also down in the bottom of the crypts of Lieberkühn are the presence of the Paneth cells, which produce their secretory product in the form of lysosome. Finally, in terms of histology, this picture is a distinct view of the section through the duodenum, and the reason we know that, certainly we can see the viliated columnar epithelium with the intermingled with the crypts of Lieberkühn, but what tells us this is a duodenum section is certainly by the presence of the prominent submucosal Brunner's glands. And again, we raised the point about what's innervating the smooth muscle down here that in this, both the longitudinal and circular layers, and the innervation is provided by a myenteric plexus, and again, as we saw in smooth muscle in the esophagus, the entire foregut, including the portion of the duodenum up to the entrance of the common bile duct, is being innervated parasympathetically by the fibers in the parasympathetic axons in the vagus nerve. This innervation also will carry out through the length of the midgut as well, basically the changeover occurring 2/3 of the way across the transverse colon, where the parasympathetic innervation of smooth muscle will be taken over by pelvic splanchnic nerves, sacral outflow of S234. Let's move down to an actual histologic section. We can see again quite dramatically a view of the duodenum. You can see again the villi and crypts of Lieberkühn, but the underlying submucosal glands of Brunner give this away as being a prominent section through the duodenum. Let's move now to a section in the notes that illustrates a section through the ileum, and the reason we know it's ileum is not, again, by the presence of the villi and the crypts, but by the presence of prominent patches of lymphatic tissue containing B cells and the related germinal centers in close proximity to the luminal epithelium. And even though we can't distinguish it histologically, that luminal epithelium is characterized by the presence of the microfold or dome cells that are the antigen-presenting cells that are kind of intermingled among the adjacent columnar enterocytes at the level of the Peyer's patches. In our next slide, let's take a view of and talk about the functional organization of the liver. And fundamentally, we're looking now at a section through a liver lobule. Basically, the organization that we want to talk about is the differences in the direction of blood versus bile flow through a hexagonal liver lobule. First off, at each of the 6 points of the hexagon of this liver lobule is what we call a portal triad, whereby there are 3 tubular structures. 2 of them are vascular. 1 of them basically are receiving all the tributaries of bile canaliculi, delivering bile to the perimeter of each point of the lobule. So the 2 vascular structures at each of the portal triads are a tributary, a branch of the hepatic portal vein, and obviously a branch of the hepatic artery, delivering either poorly oxygenated, nutrient-rich blood to the liver or oxygenated blood to the liver in the form of coming from the hepatic artery. Certainly the portal vein is delivering the dominant source of blood supply to the liver. In the range of about 70% of liver blood supply is coming through that vascular structure. The remaining 30%, in this case of arterial blood, is being delivered to the liver by way of the hepatic artery. Note that blood is flowing from each of the perimeter points of the hexagon toward the central vein, which is basically draining the blood out of each of the liver lobules, and is receiving blood obviously from each of the 6 points of the hexagon. So blood is flowing from the perimeter of the hexagon toward the central vein in the direction of the blue arrow. In contrast, bile that's being produced by liver hepatocytes and is basically being collected in bile canaliculi between adjacent hepatocytes is basically being collected and delivered toward the perimeter of each point of the hexagon. So the point is, bile is flowing in a direction that is opposite to that of blood flow. So in the inset diagram on the right side of this page, you can see a liver sinusoid, basically that's lined by a discontinuous sinusoidal endothelium. Inside the lumen of that sinusoid, we find the Kupffer cells, the macrophages associated with the liver. And between those sinusoidal endothelial cells and the luminal surface of each of the plates of hepatocytes is the space of Dease. The space of Dease is basically where all of the endocrine-based products of a hepatocyte are released into the bloodstream. They would be albumin, prothrombin, and so on and so forth, proteins. Whereas you can see the location of bile canaliculi not associated at all with the liver sinusoids. The canaliculi, as you can see, are associated with small channels between the adjacent hepatocytes into which bile is secreted. And certainly if bile is being secreted and moving in the direction of the black arrow, as we've indicated, that blood flow would be moving in the opposite direction. So, liver hepatocytes basically are the marginal functional cell type that is associated with the liver. These are highly regenerative cells. About as 90% of them can be replaced in a very short period of time, basically as little as 2 weeks. These are linked together by prominent series of gap junctions and basically separated in part by the bile canaliculi that is the site where basically the exocrine product, bile, is being produced. We've already pointed out that the microvilliated free surface of the hepatocytes are exposed to the space of Dease, and it is into the space of Dease that the so-called endocrine products of a liver hepatocyte, mainly in the form of large proteins, are released into the bloodstream. Basically, hepatocytes have abundant glycogen that's being delivered to them from the portal system that's been absorbed out of the bloodstream, prominent mitochondria, that are characteristic typically of a liver cell. And as we've indicated, the major proteins bound for export into the bloodstream are large proteins, albumin, prothrombin, fibrinogen, so on and so forth. Those are the endocrine products. The exocrine products, certainly of the liver, is associated with bile, and certainly a complete course in biochemistry could be dominated alone by what some of the vast functions of the hepatocyte are. The liver, as we said, basically contains the sinusoids that are lined by a fenestrated capillary endothelium. Inside that endothelium, inside the lumen of the sinusoid, are the phagocytic Kupffer cells, which are part of the whole overall mononuclear phagocyte system. Their job overall is to phagocytize red blood cells. So in essence, if a patient has undergone a spleen removal because of hereditary spherocytosis, the liver Kupffer cells can kind of take over many of the functions of the red pulp associated with a defective spleen. A key cell type that is found in the space of Dease are the Ito cells, or the lipocytes. Their job basically is to store fats and vitamin A, but as we see in patients that develop cirrhosis of the liver, basically late-stage manifestation of chronic alcohol abuse will cause those lipocytes to begin to secrete collagen, and that collagen basically ultimately will block up the space of Dease and the liver sinusoids and alter portal venous blood flow through those sinusoids. Now, on this chart, on this page, we can illustrate basically another feature about how the liver is organized. So we know that the liver lobule is organized into a blood and bile flow diagram. We basically can subdivide the regions of a liver lobule into zones. Zone 1, as illustrated here, is closest to the point of entry of blood to the perimeter of the lobule. Zone 2 is intermediate. Zone 3 is furthest away from the entry point of blood and is certainly closest to the central vein. So functionally, we can associate different parts of the liver according to these zones as being first or last affected by either drugs or anoxia, respectively. For example, patients that are associated with chronic alcohol abuse tend to develop signs of a fatty liver, and this basically characteristically affects zone 3 first, as well as certainly anoxia, sudden decrease in arterial blood supply of the liver, will affect the zone of the liver furthest away from the entering blood, and that's also going to be zone 3. Certainly drugs are going to affect hepatocytes in zone 1 first, because they're going to be the first region of hepatocytes that are going to be affected by those entering drugs. And then finally, let's look briefly at the associated glands that are part of the gastrointestinal system. We know that there are 3 different kinds of salivary glands: parotid, submandibular, and sublingual, having different levels of serous versus mucus types of activity. The parotid gland, high amylase activity, is a pure serous gland, whereas the submandibular and sublingual glands are mixed glands, submandibular being mainly serous, sublingual gland being mainly mucus. And we know that their cranial nerve secretory secretomotor innervation is slightly different. The parotid gland is under secretomotor control by parasympathetic axons in the glossopharyngeal nerve, whereas the submandibular and sublingual glands are under parasympathetic innervation by cranial nerve.